Pero no los quiere de sonido, por eso quiero meter cualquiera da igual.
¿Sonido? Sí, 1, 2, 3, probando. 1, 2, 3, probando. Sonido, micrófono, 5, probando. Audio, probando. 1, 2, 3, 1, 2, 3. Prueba de sonido. 1, 2, 3. Sonido. 1, 2, 3, probando.
Oye, ya pasaron los papeles. Igual que las a ver, Toño. ¿Ya?
Toño. Bien. 1, 2. ¿Lista?
Bueno, estamos a punto de comenzar. Les pedimos por favor que tomen sus lugares. Les recordamos también que pueden votar por la fotografía más popular en la mesa que se encuentra afuera. Pueden encontrar los papelitos para que después de que visiten la exposición voten por alguna de las fotografías. Les pedimos a los que se encuentren sentados en los costados que ocupen las sillas. Por favor, Tini. Carla, Víctor. Inga. Bueno, pues ahora sí vamos a darle la bienvenida al doctor Ryan Carsten. Agradecemos haber aceptado nuestra invitación y pues las investigaciones del doctor se centran en el uso de herramientas computacionales para entender la diversidad biológica. Es investigador en la Universidad del Estado de Ohio, en Estados Unidos. El objetivo de sus investigaciones es generar modelos que puedan describir la historia demográfica de acuerdo a datos reales colectados en el campo y de esta forma entender cómo las fuerzas evolutivas actúan dentro entre poblaciones para producir patrones macro o microevolutivos. Les pido que le demos la bienvenida al doctor Carstens con un aplauso.
Well, thank you for the introduction. I think you were saying nice things. My Spanish is bad, so I'm going to try to talk slow and make the ideas that I talk about easy to understand. And I am really happy to be here, and I'd like to thank the organizers for organizing this meeting. We've had a wonderful time so far, and it's just a real pleasure to listen to these talks. I've been wanting to see John Davis give a talk about biogeography for like 15 or 20 years, so this is really an exciting day for me. So what I would like to talk about today is research that we're doing in our lab. And like a lot of the work that we do in our lab, it's research that combines empirical systems of real organisms that we're really interested in with methodological work. And often our work is motivated because we feel unsatisfied with the tools that we have available for data analysis. And so I'd like to start by just briefly introducing the empirical system, and just to give you sort of a framework of what kinds of things we're interested in. One of my students, my PhD students, Ariana Marlins Garcia, who's Mexican and worked for Daniel Pinero. Maybe you might know her, she's a fabulous student. Her and I are very interested in bats in general, and in particular in myotis bats. And so if we believe the most recent phylogeny, myotis bats, the Nearctic clade is a monophyletic clade. And in particular, a lot of very common bats in North America, Myotis pacificus, Myotis evotus, Myotis lycinodes, are all closely related. And there have been a lot of questions about the species status of many of the subspecies of these bats, and a lot of questions about how they're related to one another. And one of the first things that I did when I got a lab of my own, and I had a little bit of money that I could invest in the project, was to collect data from a bunch of samples that my collaborator, Connie Dewey, and I had collected. And so I'm going to start with the data because I think that's not very interesting. And I just want to show it to you to prove that I actually have some data. So there you go. And so what these are gene trees from a bunch of samples from different species. One of them is mitochondrial, and the rest are nuclear. They're apparently non-coding anonymous nuclear members. And unlike many mitochondrial data sets, these mitochondrial data don't tell a story that seems to make sense. If we take them at face value, they imply that one of the described species is paraphyletic, and that some of that species is more closely related to a completely different clade of plants. And we don't necessarily, the reason we collected multiple data from multiple loci is we didn't want to take those mitochondrial data at face value. And so one of the things that I did in the past, and I'm going to use this as a way to start talking about the ideas in my talk, is to develop a method for species limitation that utilizes a coalescent model to try to identify independently evolving evolutionary lineages. And this is germane in groups like Myotis pacificus and their allies, where you have described species, and so the nominal species are in the blue letters. And within many of these species, there are multiple described subspecies. And taxonomically, that biologists have looked at the geographic variation within these nominal species and argued that it was substantial enough to elevate certain geographic populations to subspecies status. And so one of the questions that Tanya and I had was whether these species represented evolutionary lineages or whether the subspecies within the various species represented the evolutionary lineages. And what we did is, this work was done in 2008, 2009. Species tree methods were becoming very popular at this time. And we use the method that my friend Laura Kabaka developed called STEM. The interesting thing about STEM is that rather than being a very complex Bayesian model or estimating parameters in some way, STEM basically calculates the log likelihood of the model that you use given the data that you have using an analytical solution. And so what this means is that it's fast and that you don't have to worry about things like prior distributions and whether your Markov chains would run long enough and so on and so forth. And what we wanted to do with STEM is we wanted to calculate the probability of two very different kinds of phylogenies. One of the phylogenies where we treated these nominal species as the terminal units, and a second phylogeny where we treated the nominal subspecies as terminal units. And I've shown that here on the slide. So here's the phylogeny where all these are subspecies on the tips, and here's the phylogeny where the nominal species. So it's the same data. Here, these tips have more individuals in them than they will here. Okay. And you can see this changes the probability quite a bit. And on the basis of this comparison, it's clear that the tree on the bottom is a better fit to the data, right? It has a higher probability. The interesting thing, so I was ready to stop here until we started talking about it and realized that these were just two ends of the continuum because we had multiple species, there were multiple subspecies within each of these. And when you look at this tree, for example, some of these nominal species have basically no branch length leading to their subspecies. And others, like Lucificus and Abotus, are actually paraphyletic, and they have a fair bit of branch length. And so it's possible when we look at these two extremes that neither one of them represents the best fit to the data. There's actually 148 models that collapse some of these branches, but not all of them. There's 148 different ways that you can collapse two or more subspecies and calculate the likelihood of the species tree. And so what we proposed was to generate all of these probabilities, and because we're using an analytical solution, we can do that in a feasible amount of time. But then to evaluate these probabilities using a type of statistics called information theory. And what this allows us to do is, rather than using something like a log likelihood ratio test, which we could do if we just had two models like we did here, we can compute the probability of each model, turn that into an aggregate information criteria score, and then evaluate the relative proportion of model space that is supported by a given model. And as you can see here, we have lots of these models, and you can't make much sense out of them because they're so small. But the really interesting thing is that four of these models have almost all of the total model probability given the data. And so here's a case where if we add the numbers in these last columns, it adds up to over 96%. And all of these models are very similar. So they all treat subspecies within Myotis abotis and Myotis lucificus as independent evolutionary lineages. And the differences in models are due to a pretending variable. Some of the models, two of the Thyssinodes subspecies are collapsed, and the third one isn't, and then one of them are all collapsed. But there's no real difference here other than the difference that's caused by the penalty we're giving model complexity. So essentially, these four models are equally good, and they tell us essentially the same thing. And what this strongly implies is that we have multiple independent evolutionary lineages within what are currently described as species. Now, we didn't go so far in our 2010 paper because I had a couple of nagging doubts about this and whether or not this approach was necessarily as optimal as it could be. And since then, some of these doubts have been supported by other researchers, and some of them have been supported by what I'm about to show you. So I'll start with what other people have said. There's a very good paper by Arley Camargo that was published in 2012. And Arley looked at several methods for species delimitation. And you can summarize the paper like this. This ABC method that he used is the best. Yang and Ronaldo's DPP method is almost as good as the ABC method. And then our method is not as good. And so, I think these are reasonable results given the data that we put into it. I will say, however, that we get the same results with our Myotis data regardless of which of these methods that we use. So for example, here is the tree using BPP, and we're delimiting these Pacificus and Evotus subspecies with very high posterior probability. So what I'm sort of presenting to you is that if we use a species tree model and these newer coalescent-based methods for species delimitation, we find with very high probability that lineages within 2 of these described species are evolutionary independent. And this is interesting, and in general, I think we are currently living in a very challenging time for phylogeographic research. And the reason for this is we have an absolute surplus of models that we can use to analyze our data. So just for this phylogenetic question, we also published a tree where we had concatenated the data. So this is a different model than the species tree model. It gives us a different result. We could use other models that are coalescent-based, like INA, Jody Hayes' program, like MigrateN, Peter Beardley's program, and there's a long list. And one of the things that I like to do is analyze data with a bunch of different programs, in part because I'm nosy, and I like to see how these programs work, and in part because I have this idea that if there's signal in our data, we should see consistent results when we analyze the same data set using different programs. I think this is what most of us would like to believe. Can I have a show of hands if you would like to believe that you get the same basic result when you use different methods? This would make our jobs easy. And so I also suspect that this is not the case for a number of reasons. But importantly, all of the models that we use make different sets of assumptions. And none of the models that we use are likely as flexible and as comprehensive as any of us would like. And so we're forced to decide what processes we think are important by virtue of which models we choose to analyze our data. So when I analyze my data with the species tree program, it has several assumptions. Some of these might be perfectly reasonable given our data, others might not be. Some of the assumptions were that theta is constant and that the data are evolving in a manner that's consistent with the molecular clock. This is an assumption of all coalescent methods. Also that the shared polymorphism that we see in our data results from incompletely sorted ancestral polymorphism rather than something like gene flow. And this is something that any species tree model will assume. And so if you choose to analyze data with the species tree model, you're in effect saying gene flow either isn't occurring or I don't want to know about gene flow. And these are the choices that we're making. And in fact, we have good reason to think that trying to estimate species phylogenese in the presence of gene flow causes our estimates to not be as good as they would otherwise be. So, for example, we know that gene flow will decrease the accuracy of phylogeny estimation. Andrew Eckert and I published a paper that pointed this out in 2008. Adam Lachey has recently published a more thorough, more recent paper using the star-beast method. And there are good reasons for this. Anytime you have polymorphism that's shared because 2 lineages are exchanging alleles, and if your program is interpreting that polymorphism as being a result of incompletely sorted ancestral polymorphism, you could either get poor estimates of your topology, poor estimates of your divergence timing, or some combination of the 2. And luckily, I work in bats, and bats are a group that there's no reason to expect that there would be gene flow because they're not very mobile, right? And their distribution is such that we have these nicely separated allopatric populations. So I'm lying, right? Very clearly. So here's the distribution of the bats that our admin and I are working on. Lucificus is all throughout North America. There are these western subspecies that all come into secondary contact. And moreover, there's several different phenology studies that have looked at the distributions of these subspecies and found more or less different results. So these boundaries have a lot of error associated with them. And if I were to actually draw them faithfully, there would be these big fuzzy areas that might look like hybrid zones. And so the question is, are these lineages exchanging alleles? And if they are, is this influencing or preventing us from doing the things that we would like to do? And so how do we actually determine if gene flow is occurring? This is, I think, a surprisingly difficult challenge. There are very good programs that we can use, like MigrateN, to estimate rates of gene flow. Right? And so, for example, MigrateN uses something called an N-island model to estimate the genetic diversity and the rate of gene sharing among some number of lineages. And so here's a schematic of what this model does. you have 4 populations, and you can estimate migration from any population going to any other population, or from that population going back to the first one. And when we do this with our data, what we find is that we estimate rates of gene flow, and we get these numbers, and some are bigger than others. And so, for example, we have relatively low rates between Leucippicus delictus and Leucippicus carissima, which is good because the limitation would imply that these are separate. But between other species, or other subspecies, these rates can be much higher. So let's see, so here's a higher one. Others, they're low. These are all shown in basically per site migration rates, so that the numbers look small. But the point is, I don't have an intuitive feeling for whether these values are high or not. And I think it's really hard to know what the parameters that you get out of these programs really mean in terms of the biology of the system. Now, recently, Migrate has had a fabulous advance. So I think this is a great example of why we should keep giving program developers grant money, and over time, their programs get more refined and more useful. So in 2010, Peter Beerley published an extension that allows us to do model selection within the Migrate framework. And so instead of just estimating this n-dimensional migration matrix, we can now turn on or turn off gene flow among certain populations. And we can ask very detailed questions. And so, for example, shown here in this blue population, the genes are being shared in 2 directions. And in the green and the purple, genes are only flowing from one population to the other. And what we can do now with Migrate is we can actually evaluate which of these models is the best fit to the data. And so I've done this with our meiosis data. And what I found was that the model that has the best fit to the data is one where 3 of the 4 subspecies are exchanging alleles at some rate. And then the 4th subspecies is isolated. And so this is a result that is, of course, biologically very different than the result that we get out of the species delimitation analysis. And I'd like you just to think about that for a second. So we've used 2 methods of analyzing our data. Both are peer-reviewed, commonly used methods. It's the same data. And we would make very different interpretations just on the basis of which of these 2 methods we really thought was the right one to use for our data. So if we choose a species tree delimitation approach, we would then infer that each of these subspecies within Myosicypicus is an independently evolving evolutionary lineage. We would probably assume that these lineages did not share alleles. And if we chose the Migrate model to analyze our data, we would think just the opposite, that at least 3 of the 4 the described subspecies within Myosicypicus were exchanging alleles at such a rate that they were effectively part of the same evolutionary line. And these are not, we cannot reconcile these 2 different ideas. They mean something very different biologically. They have important conservation implications. These bats are being hammered by white nose syndrome right now. Their populations are declining at an alarming rate. And we really need to know these answers. But we need to not only know the answer, but we need to have some way of assessing whether the answer that we know is actually well supported given the data that we find. And I think this is the point of the talk where I'd really like to encourage everyone to think about how we analyze data in general. I think the most common patterns or approach that we take to this are to either summarize our genetic variation with statistics, and so we can do things like calculate FST or calculate summary statistics. We can also estimate parameters using a model, and I've just showed you how we would do that with Migrate. We can estimate something like genealogies using a phylogenetic model. And there are a lot of really developed methods for estimating parameters, for calculating statistics, for doing all of these things. But what happens then is that the phylogeographic researchers pick and choose from a smorgasbord of methods that are available. They use the ones that they think are best, and then they make their inferences on the basis of the methods that they choose. And because we're using the results of these analyses to understand the evolutionary history, or to infer something about the evolutionary history, when we get results like this, where we have some clear pattern, the first thing we want to do is make up a good narrative about how the data came to be this way. And this is sort of a joking thing here, but most of the time, our narrative is straightforward and it seems to be right. But I think it's important to remind ourselves that there are well-known cases where we get what looks like a very well-supported pattern, and it's wrong for some reason. And rather than stopping here, what I really have tried to do is develop methods that allow us to assess the confidence that we have in the results of our analyses. So that before we get to the stage where we're writing to the Department of Fish and Wildlife and helping them form a conservation plan or something like this, we actually have a way of assessing how much information we have on our data and how well supported our hypotheses are. We need to do this because if we interpret things in a qualitative manner, we're prone to overinterpretation. So we've all collected samples in the field. We know how much work that is. We've all collected genetic data, and we know how much work that is. And then we've all learned how to use all these software packages that often have really poor documentation, and the manuals aren't well written, and we know how much work that is. We put so much effort into what we do that we want it to mean something. And it's our natural human tendency to read more into the data than we can really justify given the level of information that we can get out of the data. This is pointed out by Knowles and Madison in their 2002 paper. There's also this phenomenon called confirmation bias that is well known in the psychology literature. And what confirmation bias says is basically that we interpret new information in light of what we think we know about a system. And so even the act of deciding which programs to use to analyze our data is an act that shows the confirmation bias that we might have. And so in my lab, we've set out to develop ways of analyzing our data that prevent us from misleading ourselves just because we think we know something that we might actually not know. And I'm going to talk about two new methods that one of them was just submitted. The paper describing one method was just submitted, and the other paper we're hoping to submit soon. And I'm going to use an analogy here to help you think about what these methods do. That's basically a clothing analogy. So some methods that we're going to describe are model checking methods. And we use these to assess the fit of the models that we're using. And so we hope to know if we have a model that's a really poor fit to our data, like David Byrne's suit here. We want to be able to tell if this model is maybe not appropriate. The other methods are model selection methods, where we're picking the one model that's the best fit for our data out of some number of choices. So like we did with migrant. And this is the equivalent to having a closet full of clothes and going in there and deciding which suit is going to fit the best. This is not my closet, by the way. I have one suit for funerals. So I'm going to start with Bayesian model checking. And what this really does is it allows us to evaluate the statistical fit of very complicated models to the data that we're using. And what we basically do with a Bayesian model checking approach is to integrate over the uncertainty in our parameter estimates by simulating from the posterior distribution. So you sometimes hear this method described as posterior predictive simulation. And so Bayesian model checking is a really useful approach for a situation like the Midas stats, when we want to check to see whether the species tree model is a good fit for our data. The other method will make use of information theory, which I briefly mentioned earlier. So this is a framework that has been developed to quantify the loss of information that occurs when we actually model a complex system. And it dates back to the 1950s to statisticians who worked for the Bell laboratories. And then Akeke in 1973 linked information theory to maximum likelihood. And basically this allows us to calculate the probability of a model given our data for any number of models, and then rank these models. And we need to do this again because we have so many different methods of analyzing our data. And these methods are fundamentally different. And so we can use migrate, we can use beast, we can use INA, and we can estimate parameters from these methods. But the parameters that we estimate are not going to be the same even if we use the same data. So as an illustration of this, here are data that were analyzed using Mary Kuhner's program Fluctuate. And with Fluctuate, you can either turn on population expansion or turn off this expansion. And when you do this, you can also estimate you can use this to estimate gene flow too. And what we find is that our parameter estimates actually change as a function of whether or not we allow population expansion to be part of the equation. And so under a model of no growth, we get high estimates of theta. Under a model of growth, we get low estimates. The same data. these models interpret the variation in the data in different ways. As we add parameters, the signal that we have in our data will be divided up over more of the parameters. Okay. So model checking, ideally, will allow us to discover if the models that we're using are appropriate for the data that we collected. And what they're really doing is model checking allows us to see if models are too, fitting or don't fit well for one reason or another, and to have some idea about why they don't fit well. And because of the way that we've set up our machinery, we can evaluate multi-species coalescent models and try to understand which part of them is not a good fit to the data. So to get into this, I'm going to briefly remind you of what we do when we do Bayesian inference. So Bayesian inference is a way of calculating the posterior probability by using the likelihood of the data, which is very similar to the likelihood that we would calculate in a standard maximum likelihood phylogenetic analysis, but conditioning it on prior opinions and prior values that we're using. And we're doing this in order to quickly explore tree space to find the region of tree space that is very probable given our data. And the output of a Bayesian phylogenetics software package is a posterior distribution of trees. Okay. And actually, John Halzenbeck, in 2002, described how you could use this posterior distribution to do posterior predictive simulation. And so the idea here is not necessarily a new one, but what Halzenbeck pointed out is that if you simulate from the posterior distribution, you would generate a predictive distribution that could then be compared to the empirical data. And so what this means, he was interested in the question of how do we justify models of sequence evolution to use in our data analysis. And what he said is we can simulate this posterior predictive distribution under a complex model like a GTR model, general time reversible, or under a model that's less complicated, like Jukes cancer, or under a model where nucleotides are constant in frequency over time. And when we do this, the empirical value here is the same, shown by the red arrow, but the predictive distribution will change. And a model that has a good fit to the data will look like the distribution on the top, where the predictive distribution is basically predicting really well what kinds of empirical values we see. You'll notice here that the red arrow is right in the center of the predictive distribution. And all we've done is take this idea and adapt it to a multi-species coalescent framework. And the complicated thing about this is that the model used in Star Beast has more layers to it than a model that's used in a typical Bayesian phylogenetic software package. So we are still calculating the likelihood of the data given a particular genealogy, but now we're doing this across some number of loci. We also then need to calculate the likelihood of the genealogy given the molecular clock, because we're forcing our data to evolve in a clock-like manner. And we need to calculate the likelihood of the molecular clock and the branch lengths that we get given the species trees that we're trying to estimate. The result here is a joint distribution of the species tree given the molecular clocks and the gene trees. And last year, or this year, I don't know, it was sitting at SysFile for a long time, so I think we submitted it in 2012, it was published in 2014, so about average for SysFile. We published this paper that showed that many empirical data sets where the multi-species coalescent model is not a good fit to the data. And so we examined 25 data sets that have used Star Beast for phylogeography level work. And 4 of these had a poor fit to the species tree model. And something like 1 out of 6 loci had a poor fit to either the molecular clock or the model of sequence evolution. And so what we've done since then is we've fleshed out our R package. We've made it a little more flexible, and we've dramatically increased the speed by performing a lot of the calculations on a GPU chip, which allows us to do the predictive simulations much more rapidly. And what we can now do is simulate a bunch of predictive simulations and compare values that we calculate from our empirical data to those predictive distributions. And I hope you can see this. The red line here represents where we expect our data to be if or where we expect the summary statistics to be if our data are a good fit to the model. The reason for this is that we're predicting on average that there's no difference between a value that we can calculate from the predictive distribution and a value that we can calculate from the empirical posterior distribution. And what happens is, for the meiosis data, in almost all cases, at different loci, we have distributions that are well off of 0, which is where we would expect them to be if our model is a good fit to the data. And so it could not be easier to interpret these results. It's clearly telling us that the species tree method is not a good method to use when we analyze the meiosis data. We expect, based on the results and the magnitude of some of these, that gene flow is the reason why. But we are working now on ways to use the statistic on the far right, the genealogical sorting index, to actually quantify and to identify gene flow as the culprit for this. And so that's something that we're still working on. But having basically identified that the multi-species coalescent model is not a good fit for the data for meiosis, what we now need to do is try to understand what model we should be using to analyze. We don't think that the migrate model is a particularly good fit, but it would be nice to test that assumption. And it's very difficult to evaluate the fit of complicated phylogeographic models to empirical data sets. And there's many reasons for this, but it's a sort of complex problem, and there's a very large number of possible models. And so this is something that the other software package that we are currently working on is designed to solve. And so this software is called FREPL, and this has been developed in collaboration with Brian O'Mara at the University of Tennessee. And what Brian and I are basically trying to do with FREPL is we're trying to calculate the probability of a bunch of different phylogeographic models given our data. And so ideally, we would like to be able to explore any model that we can imagine, given our data. And we really think of FREPL as basically the equivalent of model tests for phylogeography. So everyone has used model tests in this room, I would think, if you've ever done a phylogenetic analysis. What you do is you collect your phylogenetic data, you use model tests or something like model tests to choose a model of sequence evolution. And then you use that model of sequence evolution to infer the phylogeny, or to estimate the phylogeny, or calculate the phylogeny, however you do that. And so basically, we see FREPL as being a tool that is used to decide which phylogeographic model is the right model to use to analyze your data. And so our foremost consideration has been the model selection portion of this, and only secondarily the parameter estimation. And so how does FREPL work? Basically, FREPL does what we all want to do with our phylogeographic data. It looks at a bunch of gene trees, and it tries to evaluate which demographic models are very probable given the gene trees that we've observed. And so the input data are just gene trees, the same gene trees that we all know and love. And what we do is we can collect gene trees from multiple loci. And all we need to be able to do to those trees is to assign the tips, so the alleles that we're sampling, to some number of populations. And so here's an example of that. We have 3 populations that we're interested in. We would assign the alleles to these populations. And basically what FREPL is designed to do is to try to help us identify the demographic model that is the best fit for this data. So the other thing that we do with FREPL is we give users the option of defining the maximum level of complexity that their model can have, or we can provide guidelines based on the amount of data. In general, we expect that there's a correlation between how much data you collect and how complex a model you can discriminate, but we haven't sort of proved this analytically yet. That's Brian's department. It's a hard thing to do. But so FREPL also includes a bunch of useful functions. So we could define every conceivable model if we wanted to. This is a very large number, but FREPL has functions to do it. Or we can allow users to specify a certain number of models. So when I was listening to Dr. Riddle's talk this morning, I thought there are a bunch of really awesome models about the warm deserts in North America that you could specify in FREPL, and we could test all of these. So this is the kind of, I think, really useful approach when you have pre-existing ideas. These models, and this is the part we're really happy about, they can be either isolation models, so a strict species tree. They can be migration-only models, like migrate-in, or they can actually combine these two. And this seems a little weird, but there are cases when you have good evidence for the coalescence of some of the populations in your sample. And even though you know intuitively that all of the samples have a common ancestor at some point, it could be the case that there's more evidence for gene flow between two lineages than there is for populations coalescing. And if that's the case, you would want to know that that was the situation that you had. So you can either have isolation, migration, some combination of these. You can also do population expansion models, but we haven't tested that hardly at all, so I'm not going to talk about that. But in theory, any model that you can simulate using a program like MS is a model that you could analyze with. In order to calculate the probability of these models, we need to be able to estimate parameters. And so what we do is when we decide or define the complexity of our models, we count every parameter that you're estimating and use the number of parameters that are being estimated as a proxy for how complicated models are. So if you had a maximum number of parameters of 4, you might not be able to evaluate a model that had 3 populations coalescing to a common ancestor and all exchanging genes and all having different population sizes. You would have to do models that only had 4 of those processes occur. And the reason for this is motivated by a desire to force users to think about the relationship between how much data they have and how complex a story they can support. And so it should also be pointed out that these are all easily changed by changing some of the settings of the package. And so once you have parameters for each model that you've decided, the next thing to do is to simulate a very large number of gene trees under every model that you're interested in. And so here's an example where we have a model that is basically a species tree model, but there's gene flow between the ancestral population between uniting A plus B and lineage C. And so we can code this model in MS, and then we can simulate a bunch of genealogies. And now it's just a matter of seeing how many times the genealogy we observe matches the distribution of gene trees that we're simulating. And so here's a case where the observed tree has 1 match and 40 or 50 harmonies. But what this basically is it's an approximated likelihood of your model in the data. Intuitively, we know that if we do a large number of simulations under some model and keep finding genealogies that look like our empirical data, intuitively we know that this model is close to the true evolutionary history. And so this is really no different than what we've always wanted to do intuitively, but it's a more thorough examination of the possible model space. In order to do this, we have to calculate the number of topological matches. And this can be challenging for 2 different reasons. One is that you can get different gene tree distributions for different combinations of parameter values. And so even if we have the same model, like the model shown here, we could have gene tree distributions that change as a function of how much migration there was, or how deep the time involved was, or how big the population sizes were. And so in order to produce these approximate likelihoods, we need to optimize parameter values a little bit. And after a lot of experimentation and many dead ends, we finally settled on a model optimization that uses a grid search. And so what we do is we specify the boundaries of parameter space and do a quick optimization by sort of evenly sampling throughout parameter space. The number of dimensions is equal to the dimensionality is equal to the number of parameters you're looking at, which is another reason why you might not want to try to identify models that have hundreds of parameters in them, because then you have a 100-dimensional space and it's going to take longer than you want to spend. But if you have 3 or 4 parameters, it's an achievable goal. And once you find a region of parameter space that produces data with a high probability given that particular model, you can sort of imagine that there's a slope to the likelihood of the IC values. Then you move on to the next model. And you do this process of defining a model, simulating the genealogies, optimizing the parameter values for some number of models. I'll just show 2 for here. And if you get a situation like this where one model has 5 matches to the empirical data, and the second model only has a single match, then we know that the first model is a better fit to our data because there are more matches. And there are, I think, nothing about this is magic or particularly complicated. The only thing that's what's really impressive is all of the coding that has been done, and that was all done by Brian O'Mara. So it's, I think, amazing what he's been able to do with this. We have all of our code on ArtForge, and we are in the process of getting a package released for Crayon. So with the exception of 2 small parts, this is all done in R, which makes it hopefully very easy for people to use. The output that you get is basically a list of models. And the thing that I don't like is we just have numbers that code for models, and it's kind of arcane to figure out what that means, but we help try to help you figure that out. And you can then compare model fit using ISC for some of the models. Now, there are a number of challenges that we've had to overcome in order to get this to work. And these challenges are related to the complexity of what we're trying to do. We know that there's a very large number of demographic models. Even if we have 2 populations, we could easily come up with a couple hundred demographic models. And if we have 3 or 4, this number increases very dramatically. So here's a quick plot of the number of models as a function of both the number of parameters and the number of populations. And so none of these spaces are particularly big. I'm sure many of you are looking at this and saying, only 4 populations? Well, I have 5 in my current system. But it's a really complex problem. There can be millions of models, even with just 4 populations, and limiting it to 2 or 3 parameters. So the model space that we're having to explore is very challenging. And this is one reason why I personally favor an approach where you have existing biogeographic hypotheses and you're trying to evaluate these existing hypotheses. But you can use it if you have enough time on a really big computer to just churn through as many models as you can do. A second challenge is related to the space of the gene trees. And so gene trees are basically phylogenies. And we know phylogeny, the number of possible trees will increase at a greater than exponential rate as we add alleles. And so, for example, by the time we get to just over 60 tips, so 60 allele sample, we have more possible gene trees than the number of atoms in the universe. And of course, our method relies on finding how many matches under a given model we have for the data that we've observed. And so luckily, it does not take an infinitely long time to use our method, because there are several factors that we have developed to kind of get around this immense tree space. The first, I think in all fairness, we should point out that there's a difference between tree space and probable tree space. And so to illustrate this, the blue line shows a gene tree distribution that was simulated at random. And you'll notice that there's a whole bunch of trees that are all present at very low probability. But what phylogeographic data are is there's a small number of trees, small-ish number of trees that are very probable, or at least relatively probable given your data. And then this sort of approaches 0 relatively quickly. And so most of the time, the empirical data that we observe are in the probable part of gene tree space. This is why most of the time, if you collect mitochondrial data from an animal, you'll get a tree when you put it on a map that makes a lot of sense. Not all the time, but most of the time. And you have to simulate a lot to get to this part of the distribution. But the other thing that complicates this is that when we have multi-locus data, we often don't have well-resolved gene trees. We often have many individuals that share alleles. And there's all of this messiness that we can't actually resolve the true genealogies. So we've developed a number of workarounds. One of them is that we don't care about the labeling of tips within a population. So we treat them as interchangeable. And we just do a little math to average out those probabilities. So what we do essentially is we map based on any possible labeling scheme, and then we divide that probability by the number of labeling schemes that are present for the data that you put into it. The second thing we do is we allow the users to treat polytomies as soft. And so if you have gene trees with polytomies, you can basically treat those as a soft polytomy, and any simulated gene tree that would possibly match what you're observing in your empirical data would count as a portion of a match in that case. The last thing we do, and this is, I've lost my hair trying to sort this out, basically. But I think we're close to understanding what's going on. We knew that we would have to subsample for a lot of phylogeographic studies for the simple reason that if we have 200 samples, then phylogenetic tree space is so big that we would have to simulate millions of trees for each model. It would take a very long time. So what we wanted to do is pick a small number of alleles at random, work through this process, and then replicate that a number of times so that we can subsample and calculate the probability from a much smaller part of the tree space. And in theory, this has been a good idea. But it took a while to figure out the optimal level of subsampling. We're pretty convinced now that it's 4 to 5 alleles per population for a variety of reasons. But I'm not going to go into that because we don't have any real theoretical justification for it. It's just based on observations and things that we think we're being reasonable about. But we need to write a more detailed paper about that. But anyway, there's some kind of subsampling that is going to make this a much faster process because if you have a smaller phylogenetic tree space, then every single step takes much less time. The other thing that comes in handy when you have genomic scale data is that you often observe many empirical loci that have very similar genealogical patterns. And the reason for that is that you're sampling repeatedly from the probable portions of gene tree space. And so even in this example that I showed you at the beginning, some of these genealogies look fairly similar. But once you get to the point where we're collecting data using next generation sequencing methods, when we start collecting thousands of those, then we can really find that we have 50 or 60 that have matches. And then we can basically do all the calculations once and multiply it by 50. And that makes things much easier. Okay, so how does FRAPL work? What I'd like to do is give you just a few simulation testing and then show you the results of the analysis of the MIOS data. So what we've done is really a wide range of testing. I'll start with a very simple case. We can simulate data under a model that's very boring. It's just 3 populations that split at 2 time points, and there's gene flow between 2 of them. We can then take the simulated data because we know what the answer is correct, and we can ask, for some number of models, what are the model probabilities. And if it's a very simple case like I've shown here, where we just want to differentiate an in-island migration model from a species tree model from an isolation of migration model. So that's what are being shown here. Migrate in, star beast, IMA, basically. We can do that using reasonable estimates of parameter values. And in almost all cases, with a really modest number of loci, we are able to identify the true model. And so this is, I think, very encouraging. 10, 20 loci is going to be a pretty good answer. The reason that this works as well as it does is even though we're not setting out to estimate parameters well, that's never been our goal of ours. We've always looked at FRAPL as a program that tells you which models you need to use to estimate parameters. We do an okay job of parameter optimization. And so our migration rates, when migration is 0, we estimate a rate of 0. That's nice. When it's a little higher, we're usually overestimating from the true value, but not to a degree that seems all that substantial. And a similar thing gets seen with time. When we estimate divergence time, we're not quite perfect with our estimates, but we're in the general range. And this is an indication that the approximated probabilities that we're calculating are actually fairly close to what the true probabilities would be if there was an analytical way to calculate the true probabilities. Unfortunately, there's not, which is why we're doing all of this. Okay, so let's go back to MIOS stats, and I'm going to wrap it up by showing you the results of an analysis that we did where we took our data that we can analyze using migrating and estimates of gene flow, or we can analyze using star beasts or something like that, and we get delimited species. What we find is that all of our best models have a lot of similar characteristics. They all have the same topology. They all have the same. One of the 4 lineages is always involved with exchanging alleles, and it's the lineage that's right here. So it's the one that's sort of geographically in the central position. And the best model here has 40% of the total model weight. The second best model has 35%. And the third one, almost 10%. But if we add these up, that's a very high aggregate proportion. And what this is basically saying is that an isolation with gene flow model is a much better fit to our data than the other ones we explored. So I think we had something like 250 models in this analysis. So just to rephrase that, Okay, so we had 216 models considered by the analysis. The top 5 have over 98% of model probability. They all have the same topology. They all include migration, and none include a change in the population size. And so I think this is a good thing. But it's really interesting that we get the same basic phylogeny that we get with star Bs. And so even though we showed that our species 3 model was not a good fit to our data, apparently that fit didn't manifest itself in a difference in topology, but likely in a difference in divergence timing. And so Adam Lachey's paper points out that it's common for unrepresented gene flow to produce underestimates of the temporal divergence between nodes. And this looks like what we're seeing based on a really quick and dirty run of IMA, where the divergence time that we estimated using Jody Hayes' program was quite a bit less than what we estimated using StarBees. And so this paper has been almost ready to submit since June or so, but I think we're really almost ready to submit it now. And as I mentioned, the code and actually a user manual is already available on Brian Merritt's website. And so if anyone wants to check it out, I would very much like to get people's opinions about it. And in general, I'd just like to close up by encouraging you to, when you think about doing phylogeographic research, not only to worry about collecting a lot of samples and getting on a loci and doing analysis well, but pay attention to trying to figure out how well the models that you're using fit your data. Because I think we learn as much about historical processes and about evolutionary processes by evaluating the fit of our demographic models as we do from estimating parameters or these other things that we use our data for. And really, if we have a very strongly supported demographic model, that tells us many of the things we want to know about the history of the species that we're interested in. And so with that, I would like to thank NSF for supporting this work, and my past and present lab members, all of whom put up with a lot of crap from me. And I sold them some of them liberally here. So I'd be happy to take questions.
Agradecemos al doctor Casas y si alguien tiene una pregunta, es el momento. Se busca el momento. In the models you have shown to us, how are they affected by recombination? And what's the best way to reconstruct a phylogeography history if we have recombinant markers?
That's a fabulous question. So I should have mentioned and didn't that because we're using a coalescent model, we're making all of the standard assumptions of the coalescent. And one of those is that there's no recombination present. So the good news, if you have recombination, is that you all of a sudden have 2 loci instead of 1, because you can take those data and just divide them into 2. The bad news is that your estimate of the genealogy might not be as good, because instead of having 10 SNPs, you might only have 5 SNPs. But I think it's really important to check our data for that. And there are a number of methods we use. I think it's GARD, I'm not sure what it stands for. There's a way to do it in DNASP, there's a couple of other packages for it. So.
Hey, Brian. You mentioned vaguely about linking this up with the stuff that Brett was talking about. So imagine you have communities of species at the intersection of 2 of the warm deserts. Could you see taking this to the next step and seeing do you have single models fit communities of species? And I guess you can take the products of these likelihoods across them for each model and test that, but that seems like a cumbersome way to do it.
I think there's 2 ways though. So the question is, I had mentioned that, the biogeographic scenarios that Dr. Riddle was talking about could be used as models that were input by the user for Chronicle. And then Frank asked if it was possible, to somehow combine those from different species. And I think that it would be. I think that one thing we could do, all of the likelihoods that we calculate are basically calculated on a locus by locus basis. And so you could essentially develop a matrix of models by species in this case, and figure out what combinations of species were a good fit to what combination of models. And what I strongly suspect in your case, and in most of these comparative cases, is that one would find that there were several dominant historical scenarios that had support among different groups of species. And so one of the things that we often see when we run like Nickerson's in that space program is that we have more than one diversification time, but less than the number of species that we put into it. And so I would imagine that something would happen. We haven't done that yet, but one could do it. We can't even write the first paper, so it's a lot of pressure.
Right, this is probably a half-baked question. I should talk to you about over tequila later. But looking at that graph of the ever-increasing number of possible models, I'm starting to feel like, is there potentially a multiple tests problem here, like you might have when doing a genome modification study, where you're really doing thousands and thousands and thousands of hypothesis tests, or is there some reason that idea doesn't apply here?
Well, so the question is, what about multiple tests or multiple comparisons? And information theory has largely been developed to get away from that problem. And so if I understand the statistical theory behind that, and I really try my best to do that, it's not an issue because all you're measuring is the decrease in information that happens when you model the data. We know that there's some true model that we could imagine, but we can never actually do. So all we can do is sort of put the models that we can enumerate on a line between the best of those and the worst of those. And the model probabilities that come out of an information theoretic analysis are exactly equivalent to a posterior probability in a Bayesian analysis. And so the thing that we've observed that's been very encouraging is that the discriminatory ability between models is very much a function of the amount of data that you have. So when we've done model selection analyses with smallish data sets where we had 3 or 4 loci, it's very common to see several models that are all supported with 30 or 20% of the model support. And as we add loci, what tends to happen is one of those will get better and better at the expense of the others. And so even though there's a lot of models, most of them are so unbelievably bad that they have no model support whatsoever. And we can show that empirically. We can't really prove why it's happening analytically. Thank you.
Gracias, doctor Carstens. Y bueno, pues con su ponencia damos por terminado las pláticas de hoy. Los invitamos a votar por la fotografía más popular y a visitar la exposición que se encuentra a un costado del UNI. Mañana nos vemos puntualmente, por favor, para la primera ponencia.
A las 9.
Sí, sí, a las 9 de la mañana. Gracias.
Thank you.